Thank you all for coming. This is really a special Grand Rounds today. This is the 10th Calabresi Memorial Lecture in honor of Dr. Paul Calabresi, who's pictured on the board. I'm proud to welcome the Calabresi family here to the campus, and especially honored to have the opportunity to host Dr. Vincent DeVita and his daughter Elizabeth DeVita-Raeburn, this year's Calabresi lecturers. So thank you so much. Paul Calabresi is often referred to as the father of oncology, and his influence here at Yale Cancer Center remains. A former faculty member at the Yale School of Medicine, he was an internationally recognized authority on the pharmacology of anti-cancer agents. Dr. Calabresi served as director of Yale Cancer Center's advisory board until 2003. On a more personal note, I had the good fortune, or perhaps fate, to meet Paul over 30 years ago, as his son Peter, who I'll introduce in a moment, out of over 1,000 Yale undergraduates that year, ended up as my roommate. He had the top bunk, I had the bottom bunk. It was easier for him to get up there. Paul was an advisor, mentor, and friend to me, and it's meaningful to me that I now hold the job he once served in at Yale. Joining us today for a presentation on the Honorable Guido Calabresi, Dr. Calabresi's brother, and Peter Calabresi, his son. Most of you know Judge Calabresi. He's the Sterling Professor Emeritus of Law at Yale University, the former Dean of the Law School. Judge Calabresi was appointed to the United States Circuit Judge in July 1994 and continues to serve. He has received over 50 honorary degrees from universities in the United States and abroad. An expert in the care of patients with multiple sclerosis, Dr. Peter Calabresi excels in both research and clinical care at Johns Hopkins Medicine. His magnetic resonance imaging research not only expanded the understanding of the blood-brain barrier, permeability in multiple sclerosis, but also has helped in defining the timing of neuroaxonal injury, the determinant of permanent disability in this disease. Thank you both for joining us and for participating in the discussion of the book, The Death of Cancer. And I have a copy here, and we have a few copies in my office if anyone would like to come pick one up afterwards. Our guest lecturers today are co-authors of this acclaimed book, The Death of Cancer. Dr. Vincent DeVita is the Amy and Joseph Perella Professor of Medicine at the Yale Cancer Center and Professor of Epidemiology and Public Health, and he was the director of Yale Cancer Center from 1993 to 2003. He spent the early part of his career at the National Cancer Institute, and we'll hear a bit about that, of course, during the discussion today. And in 1980, the President of the United States appointed him as the director of the NCI and the National Cancer Program, a position he held until 1988. Dr. DeVita has earned international recognition for his accomplishments as a pioneer in the field of oncology. At the NCI, he developed a cure for Hodgkin's lymphoma with combination chemotherapy, proving that drugs can cure advanced cancers. He's a former president of the American Cancer Society, a Lasker Award winner with so many honors, and the co-editor of the Cancer Principles and Practice of Oncology, a textbook of cancer medicine. And I know when I was a fellow, we just said, go look it up in the DeVita. It became a household name. Elizabeth DeVita-Raeburn has written about medicine, science, and psychology for many publications. She's also the author of the book, The Empty Room, Understanding Sibling Loss. She lives in New York City with her husband, the writer Paul Raeburn, and their two sons. We're happy to have their oldest son, Henry, missing school today, and here at Yale. You can tell them you're at Yale, Henry, with us here today. Please join me in welcoming Dr. DeVita and Elizabeth to Yale Cancer Center and the Smiler Cancer Hospital, and allow us to thank them for their participation. We won't make them get up because we've got everyone perfectly situated, but we do have a small gift for both of you from the center. And it wouldn't be a Yale event if we didn't have a little picture here. So I'd like Charlie to please come up. And we also have Stephen Calabresi here, Peter's older brother. And you two are welcome if you'd like to come over. You know, Direct Connect comes out every other Friday, so we have to have some. Yeah. Thanks. Okay, now I'm going to step down. I've talked far too much, but we have really such an amazing panel, and I'm going to turn it over to the group, and they're going to have an interactive discussion. Calabresis, DeVitas, the floor is yours.
So thank you very much, Roy. On behalf of our family, we really appreciate your doing this and your organization, and I can't think of a better speaker and today to speak on behalf of my dad and his memory, and he very much valued all of what you embody, being a pioneering researcher, an educator, and a clinician. And so I'm really looking forward to the conversation today. And again, we just thank you all very much for putting this together. I thought in reading the book, which I really, really enjoyed, it would be useful for the audience to start out with dialing back to 1963, and perhaps you could set the scene for what was happening in cancer at that time, because growing up with my dad, I didn't really quite understand the battles that he was facing, and I think it was really a very different time, and we take somewhat for granted what's happened right now. So,
well, first let me say thank you to Roy for inviting us to be part of the Calabresi lecture. Paul was a very good friend of mine, and I feel certain that he'd be up there smiling down at us to have us in a panel with his brother and his son. So thank you. 1963, most of you weren't born then, and cancer was considered a death sentence. If you got cancer, you died. We had surgery, and it was not little surgery. It was very complicated, and breast cancer is a good example. The radical mastectomy was the way you treated. And a surgeon who was president of the American Cancer Society made public statements like, until we know better, everybody should have a radical mastectomy. Not seeing the problem with everybody should have a radical mastectomy, so he didn't test it against something else. So everybody got a radical mastectomy. Radiation therapy was effective, but it was very crude. So when you got radical mastectomy, you got radiation therapy to the chest afterward, and you had a disfigured chest and a swollen arm. And neither one of them changed the mortality rates, because once the tumor had escaped, and we were just about realizing at that time that there was a problem with curing cancer because of the cells that escaped the initial therapy, then you were done. So breast cancer was sort of a good picture of how the state of the art. There was very little that worked. Hormone therapy was there and was effective, but it wasn't curative. So it was a bleak field. And actually, I started out trying to be a cardiologist until I came to the Cancer Institute, and they turned me around because it was so interesting to work on the cancer drugs, and we had people like your father who was very excited about the potential for cancer drugs. So that was the environment we went into. And I went into the environment, and it was lucky for me, I went into the Cancer Institute, and I had Doctors Frarek and Fry and Zubrod at the Cancer Institute who were bound and determined that we were going to prove that we could cure advanced cancer. And so that was the environment. It was considered slightly crazy. If you were an oncologist, we didn't call them oncologists then, we called them chemotherapists, and you mentioned the word cure, you were considered on the edge, not quite in control of your senses. And it was easier at the Cancer Institute. Not totally easy, but it was easier because the environment was a protected environment. It was much more difficult at universities, which I later found out when I came to Yale. That's sort of the background of where we were then, Peter.
And what were the obstacles leading up to the war on cancer, and how did that come about? Because I think that there was a lot of people, again, as you mentioned, saying that it wasn't possible to cure cancer, and you were a champion along with some other people.
Well, the one person who believed it was possible to do it was Mary Lasker. And the war on cancer happened because of Mary Lasker. It was, the Cancer Act was passed in 1971. It was the brainchild of Mary Lasker. It was very controversial then, and it's still controversial because it approached the subject of how you spend your money for medicine. Do you spend it all on basic research or the application of the results of basic research? And Mary thought that the NIH was not focusing enough on using the science for the benefit of people. So the mandate in the Cancer Act was to support research, the application of the results of research to reduce the incidence, morbidity, and mortality from cancer. That's written into the Cancer Act. There was no timeline in the Cancer Act, but Mary Lasker made one big mistake. She didn't make many, but she made this one. She promised that if we passed the Cancer Act, we would cure cancer by the bicentennial. But since it was in 1971 in December, it was 4 years away, and everybody thought nobody would take us seriously, but they did. And it was a source of a great deal of criticism. I was director of the treatment division when 1976 came by, and we sort of held our breath, because we knew people were going to ask us. Yale, for example, got its cancer center in 1974. So we only had 2 years we barely got the laboratories built when it happened. So it was a lot of tension. And then the first Cancer Act that was passed, it was a bill, the Senate bill, headed by Senator Ted Kennedy. It had a unique structure. It proposed that the Cancer Institute be taken out of the NIH and report directly to ONB and the president. And it told the FDA drug development program, the drug approval process would be moved from the FDA into the Cancer Institute. Oh, I wish it happened. But that caused so much consternation in all over the country, NIH, academia, that another bill was ultimately passed as a compromise that gave the Cancer Institute lots of money, but still kept it in the NIH with a bypass arrangement. It was extremely controversial, and universities did not like it. They wanted the money, but they didn't want to be told, what to do. And we've lived with that controversy for a very long time.
I have a rather nasty question.
I would say, Judge, up to this
Your
time,
book describes tremendous acrimony, maltreatment in some way. When somebody came up with a new idea, as you were doing, and Paul, this was not just not accepted, but very often people were pushed aside. And I saw that very much with Paul. It then worked out very well. Now, one thinks of lawyers as being litigious, and we are, but somehow the atmosphere in a place like the law school has never been quite that way. What do you attribute it to? What is it about what is going on in medicine? Is it the money? Huge, much more money than is involved. Is it the fact that lives are at stake? What is it that creates the kind of atmosphere that you describe so wonderfully in your book?
well, let me say first, Judge Calabresi, that up until now, I've avoided being grilled by a judge. I think medicine is different because scientists are naturally skeptical, and it's good when somebody does something new. To question whether it's real or it's not real. And in the case of chemotherapy, when nitrogen mustard, which was first used here at Yale in the 40s, was put out, there was great hope for it. And in fact, the president of Memorial Sloan Kettering, Dusty Rhodes, announced that we were on our way to curing cancer. And a lot of especially the hematologists, there were no medical oncologists in those days, jumped in and used it. And when it failed, there was extreme disappointment, and they became the biggest critics. So when somebody came along like myself or Jay Freireich and proposed that we were going to cure cancer, they said, well, we've tried it, and it hasn't happened. And there was a great deal of acrimony. You're right. It wasn't the grand rounds at the Cancer Institute. Now, mind you, it was a protected environment. But the grand rounds at the Cancer Institute, when we present cases of leukemia, there used to be people from the other institutes would come up and they would shout out things like, this is a meat market, things that I had never seen happen, in any of my training. So it was a very acrimonious period. I think it stems from the natural skepticism in science, but it went beyond it in the cancer field.
I have quite a few questions about the details of the book, but before we get into that, I'm interested in the concept of the book and how you came about deciding to write it after having, of course, your textbook, which is you're most famous for, and then what Elizabeth's role was in the writing.
Yes, well, we talked about the book for about 20 years. I always thought it would be good to write a book for one main reason. The work we did was what triggered Mary Lasker to start the Cancer Act. She knew that we could do surgery, she knew we could do radiotherapy, and a patient I describe in the book, Luke Quinn, a colonel, who was hired by Mary Lasker to be a lobbyist, so she had her eyes and ears on the Hill. She was very big in the health field. She did a number of very important things. He developed a tumor in Boston, and he was operated on by Claude Welch, who was a very famous abdominal surgeon, and he was diagnosed with gallbladder cancer. And he came, they wanted me to take care of him because he was in Washington, even though he'd been treated in Boston. And I said, no, I don't take care of patients with gallbladder cancer. I only work with lymphomas at this time. And I hung up. There's Rita Kelly, you probably wouldn't know her. But about 10 minutes later, I got a call from Sidney Farber, And he said the same thing, and I said the same thing, and then he shouted into the phone in his deep voice, "You will take this patient." And said I was cocky, but I wasn't stupid. So I took the patient, and lo and behold, when I worked the patient up, it looked to me like he had a lymphoma. So he just fell into my lap. We treated him. He went into remission, and Mary Lasker was ecstatic and began to focus on leukemia and lymphomas as the missing link. And that started it. So that got me involved in the beginning. Then I later ran the Cancer Institute. And then I went to Memorial Sloan Kettering, a big private center. And then I came here. And then I was president of the American Cancer Society. And then I got cancer myself. And I said, who has all these experiences that they can put it in perspective? And so I used to sit and talk with Elizabeth. And she would prod me, and tell stories. And I would tell stories. And eventually we signed a book contract. And once that happened, we had to put the pedal to the metal and get it out. And I should say that when we finished the manuscript, it was 320,000 words, and I gave it to Elizabeth, who was a tremendous writer, and she cut it down to 110,000 words. I almost had a heart attack. She's throwing all my prose out on the floor, but it turned out when I read it, it was really, a much better book. It would have been a long, boring book originally, but she's a very great writer and brought all the color. You might want to comment on.
Well, as far as the book's genesis, these are many of the stories in the books are stories I've been hearing my entire life, and I've been hearing about Dr. Calabresi my entire life and that story. And
talk into the microphone.
And at a certain point, as a science and health journalist, I'd sit there and think, I'm sitting alone in a living room listening to these stories, and other people need to know about these stories and the people and the way that the field operates. And so, together he had the desire to tell his story, and then a desire to tell it, beyond his usual audience of doctors, that there might be other people who wanted to know. And I really had a burning desire to not be the only person who held these stories in her head. So
some of the stories in the book I wouldn't have put in initially, because Elizabeth brought them in, and she said, and the famous one is the Jay Frarek story. For those of you who have read the book, we described Jay Frarek getting drunk at a party very early in my career. Mary Kay and I were going to a party, and he was a big hero, and all of a sudden he's getting drunk and he passes out in the bathtub. And we carry him to his car, and people have said to me, especially my colleague Jean Canellis, his wife, why did you put that in there? And Elizabeth pointed out to me that, Jay Frarek, first of all, he's the hero of the book. He's done marvelous stuff, and hundreds of thousands of people are alive because of Jay Frarek. And by the way, I got a letter from Jay Frarek saying he loved the book, so he didn't complain about it. But to understand this kind of a person, he had resistance everywhere, but he had absolute determination. In fact, I have a photo of him in the book where you can look at it and you can see, just look in his eyes, you can see the kind of determination he had. It was necessary to describe how hard he worked and how hard he played when a book. So I wouldn't have done that because I'm a dull medical writer. You wouldn't write that if you're writing a paper of the history of the cancer program for a journal. But it brought people into the book, and sort of they felt like they knew him a little bit better.
it was also, what does it take to be the person who pushes the envelope in a field that doesn't always like its envelopes pushed? And so we needed to describe who that kind of person was because they're unique.
Yeah.
Perhaps you could go into the rationale for MOP therapy for lymphomas. And as I read the science behind it, I was struck with that. I think the lay community sees cancer as rapidly dividing cells, and if you have cytotoxic drugs, you can interfere with them, and it really doesn't matter what type of cancer. But really what you did was the beginning of precision medicine, and you studied the biology of different cancer cells and had a very rational approach to that, yet people still didn't believe it.
Well, it was different. It was very different from what people were doing at the time. Now, of course, we do this kind of stuff routinely. But the backbone of cancer medicine then was pharmacology. And the big problem we faced was drug resistance. It still is. We still haven't solved the problem of drug resistance. So we posited that if you use drugs in combination, if you use drugs that were individually active, you could use them in combination. And we picked vincristine, for example, instead of vinblastin, even though people thought vinblastin was better because it didn't have overlapping toxicity and we could use full doses. That was the first part of it, and that was what they did in leukemia. But in leukemia, they had a separate problem. The problem in leukemia was you just treat it until you destroy the leukemia cell, and then you wait for the marrow to recover. We had patients who had normal bone marrow, so we had to work out around how to get around it. And a new tool had come out at that time, tritiated thymidine, and we were studying the kinetics of the tumor growth with tritiated thymidine and the kinetics of marrow recovery. Jack Moxley, who worked with me, was working with Seymour Perry and was using tritiated thymidine for normal marrow, and I was doing the mouse marrow. And we laid the ultimate MOP protocol was the integration of the kinetics of marrow growth and the kinetics of tumor growth, not from Hodgkin's, but from other solid tumors, intersecting with pharmacology. So it required we felt we knew, and I think in the end it turned out to be true, that in order to cure the patients, you had to give the drugs precisely as we said you gave them, and you had to have some of every drug for every cycle. You had to give it on a very tight schedule to make sure that the tumor didn't regrow, but allow the marrow to recover. And doctors at the time were just not used to doing that. So I went around the country talking about MOP, and there's one famous story about Memorial Sloan Kettering. They had their own version where they reduced the doses, substituted a different drug, and spread the intervals, and destroyed the entire principles of the program. And they told me, well, they couldn't get it to work. Well, it was obvious why they couldn't get it to work. So our problem was getting people used to the discipline. And I think Roy would know better than I at the present time, but I still think it's a problem that doctors they see something that works and they think of their practice, and it's not practical for them to use it quite like you do, and therefore they modify the doses and the schedules to make it acceptable, not thinking that it would make a difference. The reason they don't think it would make a difference is many doctors don't believe you can cure cancer. If you don't believe you can cure, why do you want to give the patient something that makes them sick? And that was the problem. Getting people to believe you could actually cure cancer with drugs, and then if you did, then you had to be strict on how you gave the programs. It's a tough problem.
I'd like to ask you about the future. When you and Paul and others were started, it was chemotherapy, drugs that would try to kill the cancer. Recently, the move has been very much towards immunotherapy and all of that. That brings in another side of medicine, other people from the pharmacologists. And immediately there was tremendous, oh, this is it. Now in the papers, in the last few weeks, we've started reading about some unfortunate results of that, which were inevitable and so on. Do you see the same tensions that there were in your time with this new approach, or do you think that people learned from that and now are working together more?
Well, I think there are skeptics about immunotherapy today, but the hostility is not the same as it was for chemotherapy. And after all, the immune system's part of us. They're not giving foreign chemicals, so I think people are less hostile about it. And there's no such thing as a free lunch, Judge Calabresi. Aspirin, for example, wouldn't have been approved by the FDA now because it caused adenomas in lungs in the mice. So first of all, it wouldn't have been approved. And we've been spending, 60, 70 years trying to figure out how to use it, but it's pretty much a miracle drug when you think of all the things that it does. But there's always toxicity. And I think to think immunotherapy was going to escape that was not. I think we learned how to control the toxicity. I'm very excited about the prospects of immunotherapy. In fact, there's been a recent study just last week in the New England Journal of Medicine looking at a new application for immunotherapy in colorectal cancer, attacking a gene that's a very difficult gene to attack with chemicals. It turns out you can attack it immunologically. So the sky's the limit, I think, in terms of immunotherapy.
So one of the paradoxes in the book is that the very freedom to innovate, which allowed you to do what you did, also allowed others to modify your protocol in a way that made it ineffective. And I was wondering if you could comment on how you see innovation being a part of medicine and science, which is supposed to be very analytical and rigorous in some ways, yet needs to have room for creativity.
Yeah, I always say this when Roy's in the audience, that I'm speaking for myself. not for Roy or the Cancer Center. So I think the major roadblock to innovation is the Food and Drug Administration. And I think we've evened out there's less of a cliff in terms of the fall off in application of these therapies, but we've lost the peaks because we don't have the freedom to innovate that we used to have with the Food and Drug Administration. When I was starting out, one of my trainees developed a combination to prevent adenomas, which would be important in terms of preventing colon cancer. And he called me up and he said, "Tell me how you got the FDA to approve MOP." And I said, "I didn't ask them. That's how I did it." And if I'd asked them, and given the state of the drugs in MOP at the time, I estimate it would have taken 15 to 20 years to do what we did in 4 years. And that's what you're facing now. You may think that they're approving drugs faster, but every drug that they approve is approved about 2 years later than it should be. I think, for example, Opdivo and Keytruda should have been approved for general use 2 years ago. And the way we used to do it, that's been what happened. But the FDA has taken control of this. They have no business, for example, asking you to do randomized trials looking at survival as an endpoint for the approval of a drug for marketing. That's your job. It's not the FDA's job. In other words, they approve it, you test it, and you work around the issue of what the insurance companies will pay for and what you have to prove to get the things paid for. So I think in a way we're better, but in anything we're a little bit worse in terms of strictures on us by the Food and Drug Administration.
Perhaps you could talk about patient care. So my dad loved being a doctor, and I think that's one of the bonds that the two of you had, was really caring for patients. And there's several descriptions in the book that I found particularly compelling, including the young woman who had been christened neuropathy and was on a respirator and was given up for dead, and you were encouraged to keep her going, I think by Tom Fry, and of course she recovered from the neuropathy that we now know is a side effect that's reversible. And so can you comment on how long you should continue to treat someone aggressively?
Yeah, that's one of the misunderstood things in the book. Your father used to say, and I think it was true, that the secret of caring for a patient is caring for the patient. And your father was that way, and Jay Frerick was. That woman who was given an enormous dose of vincristine and was totally paralyzed, couldn't breathe, was on a respirator, I would have pulled the plug on that woman. And everybody I was working with would have pulled the plug. And we thought Jay was crazy, but we had given her a drug. We didn't know if it was reversible or not, and he wasn't going to take the chance. And after weeks, a very complicated case with fluid balance and respiratory care, she went home. She recovered and she went home. We learned a lot about recovery from vincristine toxicity, and that patient was alive last time I looked for many years. So, but people thought, well, okay, you want to keep anybody with advanced cancer alive. No, I don't. I just don't give up on patients if there's something that might be hopeful for them, that's all. And I think there are many people who will take one shot and then say that's the end of it. But I've had patients go into renal failure, who were treated for lymphomas or other kinds of cancer, go into remission and have the renal specialist come down, do a biopsy, find fibrosis, and tell me it's all over. And I've had them recover and go home. You have to be really, really careful. Paul Beeson, who is the great professor here, when I talked to him about these things, he told me that he was, sort of awakened by the fact that he had a patient who went into coma and was in coma for a long time, and they thought she was brain dead. And they didn't have as many ways of assessing that in those days. And after a month or so of being in coma, she also woke up. And he had questioned his own judgment about when you pull the plug. So if there's any uncertainty about whether you can do something good for a patient, nowadays, with all the new things coming along, you have to say, is there something that I can offer to the patient that might make a difference in their life? So I don't, if a patient's got widely metastatic cancer, there's nothing there for them. Hospice care is fine. I'm not crazy about those. Neither was Jay. Jay knew when you should stop. And I'll tell you one more story in that regard, because I've never forgotten this case. I had a patient who came in from California, and he had a diffuse large B-cell lymphoma. And we were developing what turned out to be the cure of that particular tumor at the time. We treated him, he went into remission. and he was alive more than 2 years, free of disease. And we get a call, and he's very sick. He's having pulmonary problems. And they flew in to Bethesda, and we put him in the hospital, and by this time he needed to be on a respirator. And he had round cannonball lesions in his lungs. And I was not the attending at the time, but so somebody else was managing him on the floor. But I saw him, and I looked at it, and I said, this is not what we see in lymphomas, and I just don't, we really have to be careful. We have to get a piece of tissue and find out what's going on. And his wife, lovely person, said that Ben had always said he didn't want to be put on a respirator and kept alive artificially. And she just put the pressure on the attending physician, and they would pull the plug. We got an autopsy. He had candida infection in his lung, round cannonballs, because we could treat candida in those days like duck soup. And he had no evidence of tumor at autopsy. And then, that puts the fear of God in you when you have a case like that, because this man had lots of life in front of him, and we made a mistake. It's a natural kind of thing to do, but you have to be a little bit careful about saying, I never want to be kept alive artificially if you might be kept alive.
In addition to the acute hospital care, you described providing advice to a friend with prostate cancer who went on for many years, and it seemed as if your goal was to get him aggressive therapy to get him to next year, because you never know what next year will bring. And can you describe how physicians should approach their patients and be an advocate for them in an outpatient basis, especially now when insurance companies are asking us to get on the phone and give prior authorization and approval and convince them to pay for these drugs?
Yeah, between the FDA and the insurance company, we may not need physicians anymore. I described this case in the book, and again, I didn't want to put the case of this patient, Lee, in the book, but Elizabeth told me it was a very compelling story, and we put it in, and I think she was right. And we moved him around to whatever was new at that time, and got 11 years. He had a very aggressive prostate cancer, and he lived for 11 years, very good years. And so it's an example of what you can do. Now, I have to say that in the end, I found a practicing doctor who took care of him, who was really tremendous. And how he did it, I don't know, but he could get access to drugs quicker than anybody else could, and he could get insurance companies to pay for experimental therapies. I haven't figured out how we did it, so it can be done, but it's a tedious job to do that. And I think that's what you sometimes need to do. When I got here, we had a very small transplant program. We used to transplant about 12 patients a year, and they were mostly allogeneic transplants. And when I would get patients who needed a transplant, like a lymphoma patient, I would send, from the new patient conference on Friday, I would send one up to the Farber, and then one down to the Memorial Sloan Kettering. In fact, the faculty, when they needed a transplant, usually went to the Farber. And the president of the hospital just called me up on the phone and ripped me on one side the other and said, "What are you doing, sending the patients away?" And I said, "We don't have the right program for them, and I can't keep them here just because they're here." I said, "Give us money and set up a program, and we'll do it." He did, and we have a very fine transplant program. So, I mean, you can't say, "We're at Yale, and you stay here regardless because we're Yale." It doesn't work that way in cancer field. You have to be, if you're going to be the advocate for your patient, you've got to be able to move them to where the best therapy is and let your ego, sit quietly.
You described a dramatic case in which you felt you made a mistake and pulled the plug too soon. That's rather like a case I had in which we affirmed a conviction, and then later on the person turned out to be innocent. But what about mistakes the other side? They may not be quite as dramatic. Do you feel you made some mistakes of keeping somebody alive who wanted to be excused, where it turned out really it would have been better to let that go sooner?
Yeah, I'm sure we've done that. Or at least in retrospect, you can look at it and say, we've done that. And that is a mistake. Although I think less often, at least in my experience, than the other way around. More often, people are tempted to give up than they are to prolong it, at least in this kind of an environment.
So, Elizabeth, how did you bring these stories to life and make them interesting to read about?
Well, like, as I said, I've been hearing these stories my entire life, so it was just fun to be able to tell them. And as I started to put them on the page, what I really wanted as a storyteller was to be able to, myself and for other people, to be able to see the scene. So I would pester my father. We did a lot of this by text. I would have the manuscript up in front of me, and I would text him, what color was the room, what did his face look like, what was his voice like, and he talked about someone having outrageous clothes, like, well, what do you mean, so we went pretty granular with detail to really bring those scenes to life.
I can't imagine there were any arguments or disagreements along the way.
People love to ask us, what was it like working with your father? What was it like working with your daughter? And they ask the question, and then they stand back and wait for this sort of, like, I've been in therapy for a year, But it was actually a tremendous experience because, how many people get to know their parents' life in that kind of detail and what drove them and what they were excited about in their intellectual life. And, these were stories that I grew up amongst, and I knew a lot of these people and saw them, but I understood them from a child's perspective. So as, an adult, I got to really understand my father's life and his work, and it was just a huge gift. And I didn't really know that when we started the project. I just thought he needs a co-author to help him tell this story, and I can work with him. But it just turned out to be an amazing experience.
Well, I too really enjoyed it, and I learned a lot about what my dad was going through in the 60s and 70s that I didn't fully understand, of course, as a child at the time. My dad also had cancer twice, once when I was 12 and once again towards the end when he actually succumbed. And Dr. DeVita, you had personal experience with cancer, and I think there's nothing that grounds a physician more than having the tables turned. Could you describe your experience for us?
Yeah, I think I already knew, I thought, but I found out when you're a doctor, you're no different than anybody else when you get cancer. You get this kind of a panicky sense. And it's a little bit worse, in fact, because I think I've seen just about every way a prostate cancer patient can die. And so the scenarios just went zoom, zoom, zoom through my mind, and naturally you pick the worst. So it's a great source of anxiety, and I realized that I really needed to have someone else make the decisions or help me make the decisions. And I asked my colleague Steve Rosenberg, who's a superb doctor, a surgeon, but not his field, but he's a great thinker, and let him sort of sort through the options and then present me some options, which raises a very interesting question because we often tell patients, here are your options, but we make no recommendations. And that's considered the ethical patient autonomy. The fact is it's very difficult for a patient with cancer to make that kind of a decision when they're just overwhelmed with anxiety. So to me, it was a great source of comfort for Steve to say, here are the options. If I were you, I would do this. And again, that comes in conflict with medical ethics today.
Perhaps you could provide some advice to physician scientists in the audience about if you would do anything differently or what you see as the most important principles. And especially, I found very compelling the story about when you first presented your data basically showing a cure, and it seemed as if it fell on a deaf ear. The audience was silent and no one believed you. And did you ever have doubts about what you were doing, and what was it that allowed you to persevere in the face of the public not really believing in you?
I'm not stricken with self-doubt. I don't really recall ever thinking that I was doing something wrong. I could see the data and I saw it was different. And I thank Jay Freireich particularly for that, and Paul Beeson, because when I told Paul Beeson, I was having trouble when I came to Yale because there were a lot of skeptics here at Yale. And I complained to Paul Beeson, expecting him to bring me up short. He had just decided he was leaving, so I figured if he does bring me up short, big deal. And he told me, what you saw with your own eyes is probably more valuable than what people who haven't had much experience were telling you. So I had, some call it cockiness, but I had a lot of confidence, and I didn't feel that I was doing something, that was that crazy.
When my dad, Paul's father, started out, started out in cardiology, just at the point that cardiology he thought was becoming a serious field. electrocardiography was just beginning and so on. He was very happy that Paul was going into oncology when he and you did, when that was really beginning a field, to be a field that one can. What about now? If you were starting out again, would you go back, would you go into oncology, or are there other areas of medicine that you think are today where oncology was when you started out, where cardiology was where my dad started?
Judge Calabresi didn't mention that I actually worked with his father. I did cardiac catheterizations at the West Haven VA with his father. I remember him very well. I think he probably has the gene for innovating things and passed it on to his kids. I would go into oncology. It is the most exciting field. You know, I mean, cancer is essentially developmental biology. And a lot of what we've discovered about the control of growth is basically developmental biology. The contributions to developmental biology have been enormous. I think it's the most exciting field to go into. Now, I'm biased, obviously, but I see no reason why to do something different. I just think I'd like to see more flexibility in the field to innovate so that young people can, you know, follow their ideas a little bit more. There was a review of the book by Malcolm Gladwell, and he was taken with the story I told at Memorial Hospital. Memorial took MOP and called it TOP, and they completely made a different program and didn't work. He asked me, what would I rather have? An environment that gave you the ability to do top MOPs, like the way we did, but that would also give you programs like TOPS, because other people have the ability to change it and do what they want. Or I would have less MOPs and no TOPS. And I'd rather have the environment where I have, you know, the ability to do things like MOP in today's terms and have the risk of having programs like TOP. I justify it by saying, if you look in the literature for a paper on TOP, you won't find it. They never finished the study, and they didn't do it. And if you look in the literature on MOP, you'll find thousands of papers on MOP. So I think there's no, as Winston Churchill was fond of saying, the maxim, nothing avails but perfection, may be spelled paralysis. There's no perfection in terms of testing new programs. There's always risk, and if you pull it in too tight, then you risk, you know, stifling innovation. I think, frankly, that's my biggest problem. I'm not a practicing oncologist anymore, but I would imagine I would not thrive in the current environment, given how I grew up in the field.
So maybe in the last 15 minutes we can open this up for questions from the audience, and I thought I'd start with the front row. Perhaps Dr. Fuchs or Roy or my brother would like to ask a question.
What a wonderful panel. Thank all four of you for this. So I actually have questions. I have two questions. One for the father, one for the daughter. First, Vince, akin to the question the judge had about looking back 50 years and saying, what do you see forward? If you imagine 50 years ago what you would expect oncology to be in 2016, is it consistent? Is it better? Is it not quite where you thought it would be? How would you characterize that?
I actually wrote a paper on that, and we imagined a lot of what's happening. And, you know, being the director of the Cancer Institute was a wonderful job because everything that was happening sort of crossed over your desk, and you could look at things and see the potential for it. And so we did imagine a lot of these things. I mean, Steve Rosenberg was, you know, only about 100 yards away from me and was doing things in immunology that I thought would eventually reach fruition. And it's true. He's the guy who just did that study on the RAS gene. So we thought of you could do this. We couldn't figure out why you couldn't do it. I mean, the immune system seemed to be impotent when it came to cancer until Jim Allison discovered the checkpoints, and then since then a lot of other things have fallen into place. Chemotherapy, a little disappointing because we still haven't solved the problem of drug resistance. Once cancer cells become resistant, it's very hard to overcome it. Fortunately, that's not the case with immunotherapy in most cases. I think otherwise, I think we pretty much thought these things would happen. What we didn't guess correctly was the rate of change. You tend to think they're going to occur in 5 years and they occur in 10 years. And, you know, I like to point out to people that the cancer program actually hit the mandate. 66% of $100 billion went into basic laboratory research, so support research. The application of the results of research to reduce the incidence is coming down for colorectal cancer and from other cancers. The morbidity, my God, the caring of cancer patients today is so much different. Like breast cancer, for example, morbidity, it's tough for every patient who gets it now, but the morbidity now compared to what it was 40 years ago is like night and day. And mortality, since 1990, mortality rates have been dropping overall 25%, but for some cancers, very large amounts. Colorectal, 48%, prostate cancer, 60%, leukemias, lymphomas, 80%. So, they hit the mandates. It just took a long while to do it. So Mary, who was, as the Congress referred to her, as the angel of mercy, did make one big bad mistake when she said, you'll cure it by the bicentennial. That we were never quite that optimistic. But.
Thank you. Elizabeth, in preparing the book, you obviously grew up with a lot of these stories, but were there stories when you wrote the book that surprised you, that you hadn't imagined before?
That's a tough question. At this point, all the stories have become very familiar to me. There were actually stories about my brother that I didn't know, efforts that had been made. My brother's story dealing with aplastic anemia is in the book. And there were stories about him that I found from other sources that I didn't realize all the efforts that have been made on his behalf to really try and innovate in a very tiny field to do what they could to save his life. And those things later, came to fruition for other people, too late for my brother. But so that was surprising. Many of the other stories I really had been hearing for a long, long time.
I have a question for Dr. DeVita. You mentioned that aspirin is something of a miracle drug in some respects. It clearly suppresses incidence of colorectal cancer and possibly some other cancers as well, even though it's commonplace, it's been around for a long time. I wondered if you could comment at all on what obstacle, if any, pharmaceutical companies pose with respect to treatment of cancer. Is there a tendency for pharmaceutical companies to develop medicines that are patented and prefer them over medicines that are not patented? Is that something that you want to do in any way? Is there any form legally that could be made with respect to that?
Yeah. I think in many ways pharmaceutical companies get a bad rap. You have to keep in mind that they have to make money. And if they don't make money, if a CEO of a company doesn't make a profit, the board fires him because the shareholders, who are probably the same people who are complaining about drug prices and so forth, will demand that the company turn around and make profit. So you have to understand that environment's not going to change. The biggest problem they have, I think, and I hate to keep picking on, well, I don't hate to keep picking on the FDA. Biggest problem they have is there's uncertainty in the regulatory environment. The FDA keeps saying it will use biomarkers to accelerate drug approval, but they don't tell you how they'll use them. So that they're loath to make an investment if they aren't sure how long it's going to take them to get the drug into market. The big issue nowadays is drug prices. And I was on a tweet chat, believe it or not, a while back, and I said something about competition in the market will drive the prices down. And about 1 million people jumped all over me and said it's not true. Well, it's true in most cases. But if you take a look at the kinase inhibitors for CML, they're very expensive. In fact, they keep emptying the prices. But the problem again is the FDA says you cannot market a kinase inhibitor unless it's better than the prior kinase inhibitor. If you actually let drug companies take the risk of marketing something that's very similar to this one, then they would compete and the prices would come down. So it's competition that we need rather than regulations, I think. And I think if that happened, the companies would open up and they'll make money and then everybody will be happy. The prices will come down. Very simplistic point of view, perhaps, but I think it's some truth in it.
Sometime this afternoon, the President's going to sign this bill, the 21st Century Cures, which I guess has been in the works for more than a year to provide money to the NCI, NIH, Moonshot, more support, I guess, for the FDA, be more innovative. What do you think of this? Has this gone far enough? Is it more?
I'm glad you asked that question. I was actually called by the staff of Senator Lamar Alexander, who's the chairman of the HELP Committee, Health, Education, Labor, and Pensions. And they told me the senator wanted me to know that he loved the book, and that it informed them in terms of how they were going to, propose the 21st Century Cures Act. So our book is in that act, and I like it, obviously, for that reason, and I think it will be helpful. Obviously, more money is always helpful, and we need money. There are some things in there that will help with the FDA. For example, approving a drug for a second indication. The Act says they can use real-time data without having to launch a controlled trial to compare it, against standard therapy, and that would make a big difference. Unfortunately, the FDA, I know the FDA very well, and they tend to say they will do things, but they don't necessarily follow up. And the Act has language in it that says the FDA should, and they don't say must. And I read Janet Woodcock's testimony before the House committee that was considering the bill, and it was actually very good. And then I went back and read her testimony from 10 years ago, and in terms of essentials, it was essentially the same testimony. So they have been saying for a long time they're going to do these things, and then they don't do it. What the problem, I think, and back on the FDA, I think the Act is good. The problem with the FDA is it lives in fear of making a mistake. And that's partly the Congress's fault, because when they make a mistake, the Congress jumps all over them. When they do something good, the Congress doesn't jump all over them. They don't even say anything. So what the Congress really needed to do, and there's something in the Act that may do this, is have some oversight function to see that the FDA is doing the good things they should do on a regular basis, and agree in principle, not to just argue and step all over them when they do something wrong. It's tough to be in the FDA. It's not easy.
Sure. Any questions from the audience, please?
And I have a slight hearing problem, so if you don't use the microphone, I'm just going to see your lips moving.
Thank you both for presenting your work. I just had a question about how can we get more clinicians and patients to engage in research in the real world setting? I'm just struck that among adults, only roughly 3% are participating in trials, whereas for kids, 70% are participating in trials. So at the same time, we're trying to inspire the FDA to be more innovative, push them through more quickly. After these new drugs get onto the market, how can we do a better job of really determining what works and what doesn't?
Did you say 20% adults?
No, more like 3%.
Yeah, that's 3%. So, I think patients are frightened of randomized trials, and to be honest with you, and I play this game, I shouldn't say it because I won't be able to do it anymore. If you ask people who are running a randomized trial what they think of the prospects of the experimental arm being better, they'll often say to me, geez, it's going to be better. I know it's going to be better. If you do that, it's unethical to ask a patient to be randomized to that trial. And I think we do that quite a lot. And I have an example in the book where the FDA required that after the BRAF inhibitors came out in melanoma, it required a large, expensive, randomized trial between dacarbazine, which in my experience is a worthless drug, and the BRAF inhibitor, which everybody who worked in melanoma knew was the best thing they'd seen in a long time. That was an unethical study. And I would be willing to bet that an investigator who had a relative with melanoma would never have randomized the patient to that study. So I think patients inherently sense that it isn't quite right when you say we're doing this, we don't know the result, and therefore, and the reason they don't know the result is the FDA requires a comparison for survival benefit. Okay, you have to show this, that's what they did with this study. They said you have to show survival benefit before we approve it. This is nuts, because all they had to do show that it hit the target and it worked, and then we need to worry about whatever happened in terms of remissions, how to control those emissions. So I think that's a major problem. We've never solved the problem of getting adults in a much higher frequency, but I think that's a big part of it.
Any other questions?
So you alluded to combination therapies a little bit, and I think that's really important. Complex disease like cancer, you may have one drug that hits its target and achieves its mission but doesn't cure the cancer. And so how do we go about proving that these expensive combination therapies work one patient at a time. I think it's challenging, and of course we don't want to spend a lot of time with drugs that aren't working. We want to be able to move on. So are you in favor of a utility design, or?
That's a very interesting question, because we have a dilemma. Our success has given us a dilemma. Often the new drugs, and Roy knows this to be true, when you get a targeted therapy, you give one of those targeted drugs by themselves, they have very little toxicity. But they often have no or very little therapeutic effect by themselves because they enhance the ability of another drug to work. So you can have a drug company saying, we're not going to go forward with this drug because it doesn't look that exciting. It could be, when it's used in combination, it would be one of the most exciting drugs you ever had. And I think we have proof of principles that if you want to cure advanced cancer, you're going to need combination chemotherapy or combination immunotherapy. I don't know how many times we have to relearn that, but again, the FDA requires that you test them one drug at a time. They're now asking for approvals, approval rights for combinations, and I think that's really devastating for people who want to innovate with combination chemotherapy.
I have a really important question. You're an opera lover, as am I. Why is it that in opera, the soprano is always dying of tuberculosis and not of cancer?
In the days that most of the operas were written, in the 19th century, tuberculosis was the scourge of the earth. And so that was, and in fact, composers are very clever. In Traviata, at the end of the opera, Violetta, starts at the end, she becomes ebullient and then she dies. And in tuberculosis, it was very characteristic of people with tuberculosis to have a flush prior to death and feel, for half an hour like everything was going to be all right. So the composers actually knew all about tuberculosis. And I suspect when I was growing up, you didn't talk about cancer. And I suspect it was even worse in those days. So that's why it's tuberculosis.
So we're just about out of time, and actually for those who've come to this lecture before, we always give the judge the last question, so he's had it. But I really want to thank Vince and Elizabeth and Peter and Guido for really, I think this is a very special session. I think we'll all remember this for many years to come, so thank you all very much. I'm sure they'll stay up to answer questions, and actually for the trainees, for our fellows, and actually anyone else who is a trainee who would like to join, we're going to have a special session. We're going to set up the back room for the next hour.